like to say thank you all for coming today. And secondly, I'd like to thank Papa Jack K for. I'd like to thank. I'd like to thank Papa Jack K for giving us, myself and the other K fellows, the opportunity and the resources and the funding to be able to do research over the summer. Okay, so in the past couple of years, honeybees have been a huge headline in the media. There's been cases of colony collapse disorder and just declining populations in general, which have prompted lots of bee activists and scientists and the general public to look more into it. However, I'd like to introduce you to the real star of the bee world, the yellow-faced bees, which I studied over the summer. They're also known as hylaeus. So unlike honeybees, which have stingers and produce honey, yellow-faced bees are actually small, solitary bees, and a lot of the species are actually native to Hawaii. In 2016, they were actually the first bee species to be put on the endangered list. 7 native Hawaiian species were put on the endangered list. I don't know if this is working, but they play a huge role in the native Hawaiian ecosphere and the biosphere because they are one of the main pollinators for many native Hawaiian plants, such as the Ohiolehua, Ilima, and the Ohiolehua, Naupaka, and Ilima. So why are hylaeus endangered? Well, there are multiple factors to their endangerment, but here are a few of them. One of them is invasive species such as fire ants, which have greatly decreased the population by taking over their homes, and there's just a large number of them on the island. The second one is coastal construction and erosion, which have taken away a lot of the habitat and the feeding areas of the hylaeus. There's also invasive plant species, which have also overgrown the native Hawaiian plants that hylaeus feed off of. And then finally, the factor that I am most interested in and the one that I researched, pathogens. So the virus that I focused my research on was the deformed wing virus, which is an RNA virus that's commonly found in bees, wasps, and even ants. What makes this virus so deadly is that it is carried by the Varroa destructor mite. And what this virus does to the bees is it deforms the wings, hence the name, and it also sometimes presents itself as a viral infection, which decreases the lifespan of the bee in general. So the Varroa destructor mites were introduced to Hawaii in 2007, or Hawaii and Oahu, and they completely decimated the honeybee populations. There's actually been a 67% population decrease since their introduction, and feral colonies of bees found in the wild can't even last over a year without human intervention. So a study in 2017 showed that Varroa destructor mites actually are the ones that are transmitting deforming virus to honeybees. And another study I found in 2005 showed that there's a possibility that deforming virus is actually shared through feeding off of the same flowers. So I decided to look more into that. From my research, I proposed the question, do Apis mellifera, or honeybees, transfer deforming virus to hylaeus? And I found statistics stating that 83% of foraging apis do have deforming virus on Oahu, and on the colony level, it's actually 100%. So I found that Apis mellifera were a really prominent candidate to being the ones that actually transmitted the deforming virus to yellow-faced bees. And I also looked at a study from 2010 that observed the Israeli acute paralysis virus. I don't know. Is everyone okay if I just talk out loud? Okay. So they focused on the Israeli acute paralysis virus, and they found that it was actually transmitted between honeybees and bumblebees that fed off of the same flowers, which proved intertaxid transmission. So the purpose of my experiment was to test honeybees and yellow-faced bees that were collected from the same flowers for deforming virus to prove the correlation between the two. And I hypothesized that the transmission of deforming virus does in fact occur between honeybees and non-native yellow-faced bees that feed from the same flowers. And I decided to focus my research on non-native yellow-faced bees because I didn't want to kill off the few native honeybees or native yellow-faced bees that are on Hawaii, and I would also need a lot of permits. And I thought that by doing research on non-native yellow-faced bees, my conclusions and research could apply to the native ones. So the first step of the process was bee collection. So my mentor and I went to Urban Garden Center in Pearl City, and we collected different samples of yellow-faced bees and honeybees that were found on the same bottle brush plant. We took them back to the lab, and then we froze and grinded the bee samples, and we ran them through cycles of washes and spinnings in order to get rid of all the fats and the sugars to isolate the RNA from the bees, both types of bees. Next is RT-PCR. We added a mixture of primers, enzymes, buffers, all sorts of chemicals into the RNA extractions, and then we put them into this machine, and which reverse transcribed the RNA into cDNA, which is a much more stable state, in order for us to find the presence or lack of presence of deforming virus. And that machine also amplified the DNA to make the whole process of detection a lot easier. Next, I'm sure all of you remember from bio, gel electrophoresis. So we loaded these gel trays and ran an electrical current through them, which separated the DNA according to the size of it in order to isolate the part of it that had the deforming virus. And we used different we used a fluorescent dye or a marker in order to visualize the presence or lack of presence of deforming virus. So if you can see on the top, that's one of the gel plates that we ran through, and those little fluorescent dots showed that there was deforming virus in that sample. And finally, we had a deforming virus strain test, so we took the positive honeybees and hylaeus that tested positive for deforming virus, and we ran them again by testing for specifically 3 strains of deforming virus, strain A, strain B, and strain C. And this was important because if deforming virus positive honeybees had strain A and the hylaeus had strain C or strain B, then that would show that there wasn't transmission occurring between the two. So I concluded that 23% of the hylaeus that I collected had deforming virus compared to the 100% of Apis mellifera or honeybees that had deforming virus, which was to be as expected. To put this 23% into perspective, I compared it to 2 other species of insects that have deforming virus prevalence on Oahu. One of them was the carpenter bees, and that was shown to have 27% deforming virus prevalence, which makes sense that it's similar to the yellow-faced bees because carpenter bees actually are solitary bees as well, so they don't live in a colony. And then I also compared it to the golden paper wasp, which is 45%, a much higher rate of deforming virus prevalence. But this also can be justified by the fact that the wasps are social insects, so they live in nests together, and therefore the transmission is more frequent. And then I concluded that there's definitely transmission between honeybees to yellow-faced bees. However, this transmission is pretty low. And we don't know yet if deforming virus actually negatively affects hylaeus, simply because it's only been shown to negatively affect honeybees and bumblebees. But there is the possibility that these hylaeus are actually acting as carriers for the disease and passing it on to other bees in the wild. So we should definitely look further into that, and they might actually transfer back to their nest, and it could possibly affect their larvae. Okay. So what have I learned from this K-Fellowship experience? Well, to start off, I learned how to look through scholarly articles and form research questions and hypotheses about my experiment. I also learned a lot about how to function or how to work in the lab, such as like micropipetting. I did a lot of that. And after I got my conclusions and my data, I was able to analyze it. And I also learned that you shouldn't be discouraged by your results because experiments and research is definitely a process, and in the wise words of my mentor, negative results are still good results. So even if your results don't seem to correlate with what you expected, it's still facts, and you can still use that to investigate other things. And I definitely encourage all of you to do the K Fellowship. Future research could include controlled environment testing, so it would be to more accurately prove that there is a transmission of deforming virus from honeybees to yellow-faced bees. I could also test the directionality of viral transfer, seeing if the Hylaeus are actually carriers and they're the ones that actually transmit the disease to other bees in the wild or other insects. I could also explore other means of deforming virus transmission, looking at different insects that may be sources of transmission to Hylaeus. And then finally, I could, of course, test on native Hylaeus. Okay, so I'd like to give a special thanks to Papa Jack K, of course. Jessica Santa Maria, who was my mentor through this whole thing. She was with me in the lab for hours and hours and hours every week, and played a huge part in my success in my research. And I'd also like to thank the UHB lab for letting me use their facilities and for guiding me through the whole research process, and for the moral support, of course. And then Dr. Chan, who was my advisor, who kept me on track with deadlines and answered any questions I had about the whole thing. Yes, any questions?